Picture the scene. It's a beautiful new morning, the birds are singing, the sun is shining. But you can't taste your breakfast. And you've got a dry cough. And a fever. Uh-oh, you think. Time to get a COVID test. But wait, how do they actually work? And how accurate are they really? Let's find out. Before I can tell you about how they test for this virus, you need to know a little bit about what they're actually looking for. This is SARS-CoV-2. That's the virus which causes COVID-19. Here is the capsid with the spike, membrane and envelope proteins embedded in it. And inside, you'll find the nucleocapsid proteins which surround RNA. You'll have heard of the spike protein. That's the one which the virus uses to get inside our body's cells and is also the target of many vaccines. The part we're most interested in is the RNA. That's what the lab-based PCR tests are looking for. It's the virus's equivalent to our DNA, storing the instructions on how to make a new virus. In particular, this RNA is single-stranded, positive sense RNA. Once you've taken this test, your sample is sent to a lab, where, in most cases, a real-time reverse transcription polymerase chain reaction assay is carried out. That's a horrifically complicated name, but it does describe it nicely. We'll start with reverse transcription. SARS-CoV-2 is a single-stranded RNA virus, but this test requires double-stranded DNA to work. The RNA is extracted from the sample and placed into a PCR machine. This first step uses the enzyme reverse transcriptase to reverse transcribe the RNA into cDNA, or complementary DNA. Then comes the polymerase chain reaction, or PCR. It replicates the DNA many times over, so we've got enough of it to detect. This is done in 3 stages: denaturation, annealing, and extension. More horrible scientific words I know, but this will all make sense in the end. Denaturation is the process of breaking the hydrogen bonds that hold 2 DNA strands together, leaving us with 2 separated strands. We do this by heating up the reaction mixture very quickly, to about 95 degrees C. This leaves us with 2 separate DNA strands, ready for the next process. Annealing is the process of 2 primers binding to the DNA strands. These mark out the DNA to be replicated, giving us a target to aim for. Extension can only happen in the 5' to 3' direction, so the forward primer binds to the start of the template strand, and the reverse primer binds to the end of the coding strand. This step is most important to make sure the DNA we're replicating is from SARS-CoV-2. Those primers are very specific to genes from the virus we're looking for. If the DNA we've got here isn't from that specific virus, this step won't take place, the next step won't take place, and all we end up with is a tiny bit of very hot DNA. If annealing is successful, extension is the process of replicating the rest of the DNA. This is done with a DNA polymerase enzyme, like it is in our body. But unlike in our body, this enzyme has just been heated up to 95 degrees. As you may have guessed, humans don't react very well to that kind of temperature, and neither do our enzymes, which have all denatured and stopped functioning. So we use an enzyme called Taq polymerase from the bacterium Thermus aquaticus. From its inventive name, you may be able to guess where it lives. Because the hot water vents it lives in are so hot, its enzyme should be naturally heat resistant. And indeed, Taq polymerase has a half-life of 40 minutes at 95 degrees, more than enough for our purposes. Taq polymerase picks up from the primers and duplicates the rest of the strands. The cycle then repeats. The DNA splits up, primers bind, polymerase extends. DNA splits up, primers bind, polymerase extends. We repeat this cycle about 45 times. Each cycle doubles the amount of DNA present, meaning that if we start with just 3 copies of DNA and run 45 cycles of PCR, we'll end up with over 105 trillion copies of that DNA. That means PCR is incredibly sensitive, needing just a few copies of the original RNA to work. Finally, the real-time part of the name tells us how we work out whether the test was positive or negative. To simplify things, I'm going to talk about just one PCR kit, which is used here in the UK. Not all real-time PCR works the same way, but the principles remain the same. It's called a TaqMan assay, which is about as fun as biological wordplay gets. It uses a TACMAN probe made up of 3 major components. This F is the fluorescent dye. When you shine a particular wavelength of light at it, it will absorb it and emit a different wavelength back. Next, the DNA in the middle, which is complementary to one of the genes being replicated. Finally, the Q is called a quencher molecule. It blocks the fluorescent signal when it's close to the dye, which it is here. When the Taq polymerase begins to extend the strand from the primer, it also breaks apart any DNA which is in front of it. When it gets to the probe, that breaks it up, separating the fluorescent dye from the quencher molecule, and allowing us to detect the fluorescent signal from the dye. As this process repeats with each cycle, more dye is released, and the fluorescent signal gets stronger, which tells us the result is positive. The TACMAN assay works kind of like Pac-Man, which is where it gets its name from. The Pac-Man eats up the probe in front of it, allowing us to receive a signal by separating the fluorescent and quencher molecules. This can only happen if A, the primers are complementary to the target DNA, and B, the TACMAN probe is complementary to the target DNA. It adds another check to make sure the DNA being amplified is definitely originally from SARS-CoV-2. This kind of PCR test is called real-time because you can detect the replication while it's happening and produce a nice graph. Of course, PCR tests aren't the only way to test for COVID-19, but they're often called the most accurate. There are 2 important numbers to look out for when considering how accurate a particular test is. That's sensitivity and specificity. Sensitivity tells you what percentage of people with the disease will return a positive result. So, if a test sensitivity is 70% and you test 10 people who you know have COVID-19, then 7 tests will come back positive and 3 negative. This tells you how many false negatives you get, so how many people test negative but are actually positive. A high sensitivity means a low number of false negatives. Specificity tells you what percentage of people who return a positive result actually have the disease. So, if a test specificity is 80% and you get 10 positive results, you can expect 8 of those people to have COVID-19. Specificity allows you to work out how many tests will return false positives, the number of people who test positive but are actually negative. The ideal test would have both 100% sensitivity and specificity, so you'd get 0 false positives or negatives. PCR tests have a sensitivity of about 95% and a specificity of almost 100%. That means that if you get a positive result from a PCR test, you almost certainly got COVID-19. If you get a negative result, there's a 95% chance you don't have COVID-19. But for the most part, what the PCR test says either way is considered correct. The most used alternative to PCR tests are lateral flow devices, or LFDs. In the UK, we use Innova LFDs, which are rebranded under the NHS logo. There are varying numbers for their accuracy, but in a recent analysis of government data, they were found to have a sensitivity of 50.1% and a specificity of 99.97%. That means they'll catch about half of all cases were you to test the entire population. They're also very specific, so false positives are rare. In fact, if you carried out 10,000 tests, only 3 would be false positives. Now that makes them sound very accurate indeed. However, that statistic isn't quite representative of the real-world consequences. As I record this video, 1 in 80 people in England have the virus. That's about 700,000 people. Of those, the lateral flow test will pick up half, so 350,000 people will be expected to test positive. Those are the true positive results, with the remaining 350,000 people returning false negative results. Of the other 55 million negative cases, we said earlier that 99.97% of them will test negative as a true negative result. That leaves about 16,000 people as false positives, which is a lot, particularly if they then have to self-isolate. In total, 367,000 people will have to self-isolate, of which 5% will be self-isolating for no reason. Okay, well, that's not great, and a lot bigger than the initial number seemed, but it's not that bad. This gets a lot worse when COVID-19 cases are low. In the lowest point of the summer, 1 in 2,300 people had COVID-19, so 24,000 people. Again, 12,000 people will be true positives, with another 12,000 being false negatives. Of the remaining negative cases, 16,000 people will be false positives. That's more false positives than true positives. 58% of people self-isolating will be self-isolating for no good reason. And that, in a nutshell, is one of the reasons why we don't use lateral flow tests to determine who should self-isolate. It's also a very good example of how equally valid statistics can be manipulated to tell very different stories. Of course, that's not to say you shouldn't be taking lateral flow tests. They're a good way to identify cases we wouldn't otherwise know about. But it's also important to remember that a negative result far from guarantees that you're virus-free. That's it for this video. Let me know what you'd like to see next. Please do like and subscribe, and I'll see you in the next one.